Thank you. Well, we've come full circle. Started with research and ending with research. As a research scientist, I think that's the way it should be. But I'll try to keep you awake a little bit. We're going to run through some research that's currently ongoing in the FBI lab or with some of our partners. I'm going to try to restrict it to that, and I'll just give you a short synopsis of the projects. But the first thing I wanted to do is I want to see, by a show of applause, how many trace evidence examiners do we have in the room? Okay. How many non-trace evidence examiners? Okay, so we outweigh them. and that's a little trick I learned from a teacher friend who basically told me that if you do that at the beginning of your talk and you have people who aren't satisfied, at least you've gotten applause at that point. Okay, so I work in the Counterterrorism and Forensic Science Research Unit. For those of you that are familiar with the research that's been done at the FBI lab for nearly 30 years now, that was the Forensic Science Research Unit that was started. They've added counterterrorism and intelligence to our mission as well. And I'm going to have to step to the side a little because I can't see my teleprompter. So what we do is our mission is to formulate and execute research, not only for the FBI lab, but for the FBI as a whole, and for our federal partners and state and local requirements as well. So we are open to do research that supports the entire community. As I said, we have added counterintelligence, counterterrorism, and traditional forensic science to our mission. So with limited resources and trying to spread very thin, you could imagine that certain disciplines are given more weight than others. And we cover the research that we do, both internal and external, with a visiting scientist program where we bring people into the FBI lab to do research with us, and I'll speak a little bit about that at the very end. And then finally, we do have interagency collaborations. Again, in the time of limited resources, we are trying to coordinate federal research so we can leverage each other's abilities and capabilities, and also so we're not duplicating efforts. So as an example, we do collaborate with the NIJ. I am a member of the technical working group that defines the research needs for the NIJ and the forensic community. And I'm also a member of a DoD forensic science research group as well. So we are making some impact in coordinating the federal labs, which should be good news for the tax-paying US citizens here. So we'll start our research initiatives. I've broken them up into the various sub-disciplines. Essentially, we cover biology, which I won't cover any of that, physical sciences, and chemistry. And I've highlighted areas, materials analysis, microscopy and microanalysis as the areas I will speak about today. But that section also covers latent prints and question documents. And you could imagine that with in the last, I would say, 10 years or so, with the challenges to those disciplines in court with Daubert standards, the National Academy's report, et cetera, a large emphasis has been placed on developing and addressing the research needs of those. And I think that the areas of microanalysis have taken a back seat to that. Some of my own research work that we've been doing internally involves the application of cathodoluminescence to materials. And Chris Palahniuk was a postdoc of mine a number of years ago, and this first slide shows some of the work that he did when he was with us, where we're looking at the application of this technique to a number of materials, both geologic in origin and materials that contain geologic minerals. And so you'll see examples there. We've got multi-layered paint. This is something that Tom Hopen did years ago, where he showed that you could readily see layer structure in white paint. We've looked at concrete block, and the luminescent colors tell us what the mineralogy of the aggregate are. And then across the bottom, you see some of Chris's examples of playing with some cross sections of duct tape, and just a quick look at what we can see by cathodoluminescence, looking at various different brands of duct tape. The summary of this work is included in a book chapter in the Forensic Analysis on the Cutting Edge that was edited by Bob Blackledge a number of years ago. But this work was the precursor to some of the additional work that we've done in cathodoluminescence, more carefully looking at the characterization of not only the colors that we're seeing, but also the spectra from certain mineral types. So this is an example. We looked at feldspar minerals as an example. And we've taken samples from the Smithsonian Institute from known locations and of different feldspar type and done cathodoluminescence. If you look at the spectra that you see you'll see this top graph is microcline, bottom is labradorite, and there's 4 different sources which are mapped on the map in different colors. The spectra, don't worry about the peak heights, that's a function of orientation, but the peak positions, the peak centroids, are different within the type. And so this gives us some indication of being able to separate feldspar types by the location from which they came. This is tied to their chemistry as well as crystallographic differences. We're currently in the process of writing this up for publication. We've then also gone on to look at sediment samples. You saw some of this presented at the last Trace Evidence Symposium. We're currently working on a method to automate this as much as possible so that you can, when you look at the CL image and you can see the different colors, you can readily identify the mineral grains of interest. And I think that that's probably one of the strongest capabilities in its use for mineral identification. We've gone on to compare that to what we get with SEM imaging, SEM EDS identification of the minerals, and we're able to actually, at least for the feldspars, look at what the percent anorthite, for example, percent orthoclase would be of the feldspar mineral. This has also led into some nuclear forensics work, looking at the changes in the carbonate mineral spectra with application of neutron dose irradiation, and that is also something that's coming out for publication soon as well. So going on to glass, the SWGMAT Glass Group, I actually didn't put anything together on SWGMAT Glass because I knew Maureen was speaking before me and I assumed that she would cover that topic. But the SWGMAT Glass Group is strong, we're working hard, and we are working toward interpretation methods, and I'm sure there will be some other discussion. We're working on ASTM standards in both SWGMAT that will review them and the Elemental Analysis Working Group that's funded by the NIJ. And we also are working on some international collaborations looking at the refractive index and elemental variations both within and between glass products of modern manufacture to do some match criteria assessment. You'll hear a lot this week about the EAWG, the Elemental Analysis Working Group. As I said, it's being funded by NIJ and run out of the Florida International University. We are working under what was our night crime model, which was a European Union-funded thematic network to improve the forensic analysis of glass. And we're doing it through validation and standardization. We're looking at multiple methods of elemental analysis. We're looking at multiple instrument types within that method. So for example, different vendors, different X-ray optics, etc. Everything we're developing is going into either publications, presentations, standardized methods. This is done through round-robin exercises, which are actually interlaboratory exercises. They weren't true round-robins. But we're trying to determine a common language that we can use to explain what we mean when we have an elemental match by different methods. So the members of the EAWG, there are quite a few. There are 31 organizations currently involved with 31 members with 23 organizations. And I don't know if you can read the note there. We're taking trace evidence analysis higher. As I said, you'll see a lot about this, so I won't discuss the round robins. I believe Christine Olson will be talking a little bit about that in her presentation this week. There were 4 of them on glass. There was 1 on soil. And out of the deliverables that have come out so far, we're working on 2 publications. We have draft ASTM methods for both glass by both XRF and laser ablation ICP-MS. And we have a number of presentations here at this meeting that are really directly investigating the application of the methods, the figures of merit, and the significance of the association. Just a few quick overview slides on other types of evidence. I don't do hair analysis, and I don't do paint analysis for the FBI lab, but the Trace Evidence Unit has a number of projects that are looking at hairs and fibers. And so I've just put these up for you to speak to the people who are listed as contacts here. When you're here at the meeting, they are here. Steve Shaw is running a project on the microscopic effects of hair that are removed both antemortem and stored under various conditions to understand what characteristics could be consistent with postmortem root banding or putrid roots. Trace Evidence Unit is also looking at fabric damage in a burial environment and physical matching of fabrics and the evidential value of textile fibers. So you can go ahead and speak to Sandy or Steve about those if you have interest. Diana Wright spoke to our SWGMAT group recently about the characterization and discrimination of single white layer architectural paints, and she will be presenting that later this week as well. As far as our chemistry initiatives, most of them are directed at explosives analysis. And I don't know how many explosives analysis folks there are in the room, but I did see a session on something going boom, so I thought you'd be interested in this. Most of it is geared at novel explosives. So looking at hydrogen peroxide, trace residues of hydrogen peroxide for peroxide bombs. We have a project, this is both internal and external research, looking at metabolites in human hair to try to determine if we can assess a person's role in either making bombs or handling bomb materials. There's a project that's just getting kicked off on multidimensional GC FID for the analysis of explosives. And this is really targeting compounds for highly complex explosive samples. The first material that they are going to look at is the oil fraction of plastic explosives. But as I said, this is just at the very beginning stages. And that's handled by Chris Tipple from our group. The last explosives topic that I was going to discuss is we're looking at factory-caused isotope variation. So this is using stable isotopes in explosives and documenting the temporal variation from the specific factories that are producing the explosives. And that's Dr. Libby Stern. She's our stable isotope specialist. And any of these people would be happy to discuss the research with you. I know as a research scientist, we always want to talk about our research. And then the final area is trace volatiles analysis. This is really looking at, this is a large program area looking at the ways to assess and the actual assessment and identification of the compounds that are the components of human scent and human odor, mostly from decomposition processes, but also from live humans for trailing, tracking type purposes. Also, they are using this profile to come up with novel training aids to make the dogs perform better if they can identify the compounds which the dogs are actually hitting on when they find a buried body or when they track a human being. And that program is being run by Brian Eckenrode, who's also out of the research group. So finally, just to wrap up, we'll do a little plug of our own here. As I mentioned in the beginning, we do have a visiting scientist program, and it's managed by the research unit, and it's coordinated through a group called ORISE, which is out of the DOE. And what we do is we bring in students and postdocs, faculty who are on sabbatical perhaps, to come into the FBI lab, do research with us, and hopefully build relationships with the scientific community and with the academic community as well. But it allows the visiting scientists to not only continue their education, but to give them some real-world work experience to become trained and proficient and understand and develop a research culture, which I think is important in the forensic sciences. So the small drawback to it is that you must be a U.S. citizen, and you also are required to pass a security clearance. So everyone here should be able to do that. So you must have completed your degree, whatever your last degree was within the last 5 years in order to be eligible for the program, and or you can be affiliated as a faculty member as well. And I've put the website and all the various information up on 3 slides in case you're really interested and you were writing it down. If not, you can always get in touch with me, and we'd be happy to discuss that with you. And I encourage you, we have a number of both current visiting scientists and former visiting scientists. I encourage you, if you're interested in the program, to speak to them and find out about their experience. And I think I've done it in my time. So if there's any questions, I'd be happy to answer them at the session. Thank you.
Thank you very much. All of the panel, we appreciate it very much. Let's give them another round of applause. We invite you to come to the microphones with questions, and there's a little known technology we have in this hotel. If we don't use those microphones, your beer tickets don't get validated. So please think of some questions, and you can go back if anybody would like to ask questions of this panel regarding domain relevant information or contextual bias. They would be welcome, I'm sure. So please come to the microphone if you have questions. I'm going to have one question while many of you are gathering there for the panel. We talked about research and its use and its value and so forth. But how do we ensure that this research, applicable research and validation of methods, gets into the laboratories, particularly laboratories that may not be well connected with this group? How do we ensure that so that these standard consensus methods, research results, and so forth are used? Does anybody want to talk about that? Sure.
Sure that you're educated and that you understand the research that's going on in your own field. I mean, really, we talk about this all the time with SWGs and how do they disseminate that information? How do you get people to follow the standards that the SWGs develop and so forth? Once again, not to put DNA off again, but what DNA has that nobody else has is they have CODIS and they have just a whole different system, if you will. So they can force their laboratories and basically say to them, if you want to use CODIS, then you have to follow these standards and so forth. No other discipline really has that carrot dangling over them. So what I would submit to anybody in any of the other disciplines is that it really, I mean, the responsibility is up to us. It's up to the individuals, to gather as much information as possible. The community itself, groups like ASTE and the SWGs and so forth, they can only do so much. They can only disseminate as much information. But once again, it's up to you to continue to educate yourself and to bring the information back when you attend symposium, those types of things.
Claude.
Yeah, I agree, but I would just add something about the fact that probably at the beginning of the research, probably operational forensic scientists should be heavily involved right from the start. And there was a slide, I think, in the previous panel. It doesn't mean that the people very busy doing casework would have to do the research themselves, but at least, being involved. And I think it's the best way, when people are involved, they are more likely to take the outcomes up.
Hard enough just to operate a laboratory, let alone equip it with some of these new technologies. What do we do in that situation?
Question. JoAnn.
Okay, so I'm not going to answer Vinny's question because I don't have a budget, but I will say that as part of our research portfolio, the way our projects are managed is that we do have caseworking support or other government agency type support, so that there is an assurance of some transition into a laboratory setting. And then for the community outside the FBI, it's meetings such as this or the other professional organization meetings where we present our research, where people become informed of it, and then they can transition it into their laboratories.
We've got a question.
Yes, I'd like to kind of refer back to the a question that was asked at the first panel regarding the future of trace evidence and whether we think it's going to go extinct, and I think we all agree it's not. But if I could kind of rephrase the question more focused on where do we see the future of classical light microscopy methods in trace evidence, and do we think that those methods are going to go extinct?
JoAnn.
Well, I would have to say I would hope not. As a trained microscopist, I really feel strongly that you need to do classical microscopy, and it's cheap. And once you're trained well, you can really do a good job, and you can do a lot with it. So I think that there will always be a place for it, and I would hope that as we educate community and outside of the trace evidence examiners, we educate people about what we do and the value of the trace evidence, we can show that you don't need the most expensive piece of analytical instrumentation, just like you don't need mitochondrial DNA for a quick hair exclusion in many cases. So in the right place, I don't think it will ever go out of style.
Also, I'd like to add just, well, first of all, mass spec guy, so I'd say put the light microscope away and mass spec it. But if you notice some of our NIJ training awards, and I say that facetiously, I'm an advocate of microscopy, but if you look at some of the NIJ training awards that we've done in the past 2 years, we've actually had these very same discussions. And of course, I was a drug chemist at one time where, we used to do, we used PLM for doing a DL or dextromethorphan differentiation. And we kind of had this discussions about the utilization of light microscopy. But we are funding, or we continue to fund training classes through Macron and a couple other places. So at least on our radar, we know that it's very important for the community. So we do continue to fund training in microscopy.
Yeah, I would just add that if you go back to some of the slides I presented, I mean, as I said, we could use forensic science in different levels, and I guess obviously depending on the question you ask, light microscopy is crucial because it's cheap. It can be, can give a quick answer if you have a well-trained examiner. So, I hope it won't go away. I mean, it's really absolutely crucial. And I must say, from an outsider, I think the American forensic science community has a very strong microscopy background. I mean, without denigrating my colleagues in Australia, I would say that in what we call chemical criminalistics, the equivalent of the SWGMAT, we've got the chemical criminalistics SAG. I don't think the level of microscopy is very high compared to what you have in this country. So please don't get that away.
I would just add that, like Rock Harmon said, don't make eye contact. Don't listen to those people that say, "Oh, it's just a microscope." You're just looking at things. And
I think it is important that we keep educating ourselves and the new generation of trace examiners, as well as the forensic community in general, because as accreditation standards become more strict, documentation standards become more strict, there is going to be a push to marginalize microscopy. And we need to have that conversation now before we do become marginalized.
thank you. A question?
Yeah, I just want to add one more point to this.
Sure.
Chinese, not assertive enough, I don't know. Microscopy is the soul of trace evidence, and I think if it goes away, I think so does trace evidence. From my perspective, the best practitioners in this field are excellent microscopists. I'm not a good microscopist, so you draw your own conclusion from that. But it's just an invaluable tool in this field, and I think there's just so much information you can get from it, and just from taking a look microscopically at something, the information that you're going to get from that sample, and the information that you're going to learn, and the things that you're going to learn just from looking on that small scale are just completely invaluable.
I can feel Skip squirming in his seat over here. Question?
Sorry, that's very loud. My name is Keith Inman, and for those of you who only see me as a DNA person, let me establish my bona fides and say that I had an advanced certificate in microscopy from Macron before I could spell DNA. So I speak your language a little bit. And Dick, I'd like to get back to your question, actually, about that transition or translation of research to the bench level. Both Claude and Michael referenced a paper by Jennifer Mnookin that talked about the establishment of a research culture within forensic science. And one of the recommendations in that particular paper is that the beginning of every research project basically is you. You have a problem. Something presents itself almost for the first time to you, and the question is, well, how do I deal with that? And as Vince and others up there have indicated, you have cases to get out. So a research project is basically not in your future. One of the suggestions within the article is that laboratories establish what I like to call town-gown relationships with universities. University professors are dying for things to do. Now there's always the issue about money, Gerald, and how those relationships really start. But the way that we envision this, and I think this could be a lovely model, is you have a problem. It's relatively unusual. You take it to the university individuals who have a gaggle of free labor in the form of graduate students who can do the 10,000 experiments or replicates that are actually required to answer your question. And once some of the validation that the earlier David Exline has some expertise in, once that sort of baseline validation and information is in hand, with perhaps the development of relevant databases, you get to cycle that information back to the caseworkers. And so you actually have this continuous loop of problems coming from you, the problem being addressed by, frankly, people who have both resources and backgrounds to do a very good development and developing answers or some solutions to that problem, and then it gets cycled back. And that doesn't exist anywhere in this country. I would suggest that the fact that Richard has to ask the question, how does this research get back to you? I want to sort of bring it back and say, how do the right questions get to the university people? I won't be trite and say it takes a village, but it does take everyone really understanding that there are various roles to play, and there probably is a model that we could develop that would address the needs of everyone here, certainly in terms of that bigger picture, how do we develop a research culture? And I would be remiss if I didn't say that Dave Stoney was on that paper too.
Thank you. Any comments about that from the panel? Disagreement?
I would fully agree with Keith, and I think, if you want to talk to either Sally Coulson from New Zealand or myself, and Sally, in New Zealand they did a fantastic job with local universities like the University of Auckland, and from the university side, I mean, we have a quite extensive network in Australia of research going on with various operational agencies. So yeah, I guess they're probably interesting information and experience to share.
Anything else? Sir.
Yes, Larry Barksdale, Lincoln, Nebraska. Wow, Mr. Inman. My interest. My question being, what is the feasibility in terms of the future of trace evidence for migrating some of the more traditional roles of the laboratory scientists? And I'm thinking in terms of the comparative sciences, trace evidence, hair, fiber, footwear, tire wear, blood stains, maybe QD, to the functions of the first responder. That being the law enforcement officer, the firefighter. And the basis for this, I'm thinking such as toxicology, breathalyzer analysis, environmental analysis, not only for air quality, but hazardous materials by firefighters. And I guess I also would submit that in terms of a question for a research venue in terms of modeling that may be something that would address trace evidence and its application to forensic sciences.
Anybody have a comment on that?
I think that you're going to see more on-scene examinations as we move into the future, and this isn't only with trace evidence, fingerprints, it's going to be DNA as well. I do think that it's completely essential that you have a scientist doing those examinations, not somebody who might have received a 40-hour course to run an instrument, make a determination on something. You know, I spoke about it yesterday in our workshop on general unknowns. I get a lot of stuff that gets screened by hazmat teams, and they're using these instruments that have libraries in them, and they just think because you get a library hit, that's what the compound is, and they write it down, this is what it is, and they submit it to the lab, you know, already thinking they know what it is, and then when you tell them it's something different, they don't really like that. I think you see it in drug analysis as well with some of the presumptive tests and the field testing that's going on. I think it's extremely dangerous to do something like that if the person who is not doing the examination does not have, it's essential that the person have that scientific background and understands the technology that they're utilizing and the chemistry involved. I don't know if anybody else has a comment.
Joshua Friedman from the FBI lab in Quantico. I was just curious about the panel's ideas and thoughts, particularly the NIJ's thoughts on the recent legislation that has been proposed in Congress about the National Institute of Forensic Science and standardizing procedures across all the labs.
Jerry.
Wow. Let's talk offline. In all seriousness, no, there's a lot of, there's a lot of talk right now. There's a lot of things that are happening. There's a lot of balls going in different directions. So there really is no concrete answer to what you're asking. I mean, all I can say is that, I don't know if those of you in the room are familiar with the Subcommittee on Forensic Science, which is under the Office of Science and Technology Policy, which is under the National Science, and there's all kinds of acronyms. Basically, it's under the White House. So I'm the co-chair of the Standards, Practices, and Protocols Interagency Working Group, in addition to Sue Ballou from NIST. But we've been discussing these, we've been having these discussions for really since the NAS report, but there's really no concrete answer to what you're asking.
Any thought from outside the NIJ? Question.
Hi, my name is Sarah Chu, and I'm from the Innocence Project. And I just wanted to respond a little bit to what Vincent said. I'm hoping that even though we're in this adversarial system, that the people who you might have originally thought of or historically thought of as critics, that you might see now as maybe people with high expectations. At the Innocence Project, we know that DNA isn't the answer to everything. And there are cases that we're working on where we've needed to rely on non-DNA evidence to help support a case for innocence. And so it's really important that we strengthen non-DNA forensic techniques so that they have more power to administer justice. And I'd say that what we want to see is that science is used within its parameters. And to do that and to strengthen that, we need to see more research. And we're advocating for more research funding at the federal level. And as Michael said, as people who work in the innocence movement, we just want to see the system become more accurate, which means that we support methods that help protect innocent people and help identify the real perpetrators. And that's what our concern is in this system. And what's exciting about the last few days, I feel like the distance between us isn't that large, and maybe it's 85% or 90%, because I see people who find transparency to be incredibly important. There's a desire for rigorous research, there's a desire to develop best practices, and there's a desire to improve training here. And so I'm going to be here for the rest of the week, and I hope that I'll be able to speak to more people. And if you have any ideas about how we might be able to, bridge that 15%, I'd be happy to speak to you.
Comments?
I just want to say thank you for your comment, and it's really, I'm very glad to hear that you're in support of some of these things. Some of my perceptions of some of the stuff that came out of your organization revolves around some of the articles saying that forensic science was responsible for a lot of wrongful convictions, when in fact, I think it's something like 90% of those convictions were really based on bad eyewitness testimony or jailhouse snitches. I don't know if you want to comment on that, or I'd love to talk to you later about it, because I reviewed a lot of those cases, and I think there's a lot of confirmation bias that comes in on the back end of things, where somebody either falsely confessed to something or was fingered by somebody in a bad eyewitness type situation. And I've read tons of stuff on this where people say that once a jury sees somebody say, that's the guy, I saw him do it, that they're not going back. They're not going to listen to anything else that anybody's saying. Then all of a sudden, there's an article that comes out that says, forensic science is at fault here, when it's really just such a small part of that system, and there wasn't anything that was terribly wrong said to begin with.
What we do when we get cases is we break down our cases, deconstruct them, and look at where the system went wrong. Because technically, if you're an innocent person, you shouldn't be convicted. So what we found is that there are multiple contributors that reoccur throughout the cases that we review. And among them are unvalidated forensic sciences or improperly applied forensic sciences.
I spoke about hair, and I think that's a really good example, where somebody got up there and said these hairs, compare microscopically in all physical characteristics observed. They're not saying that this is the hair from that person, but it seems like in the book, as I was reading it, that's what was being said, that this forensic scientist got on the stand and said, this is the person, we found their hair and that's it. Well, they got up there and said this hair could have come from that person or anybody with those same physical characteristics.
Well, to
We
address
could talk about this later.
one of your earlier points, we don't know how much one, like you said, if it was the eyewitness misidentification or the false confession or the forensic evidence that was the greater contributor.
I'd say if you have a false confession, there's really nothing else that's going to turn it around from that point.
But what I would like to point to is an article by Peter Neufeld and Brandon Garrett where they deconstruct testimony, and so they don't include cases where testimony was given within the parameters.
Let's don't debate the Innocence Project anymore. But Michael's got a question, and I'm sure that'll move us along.
Yes, it'll move us along. Two things. I think your point is well taken. What happens is that you get multiple contributing causes, and in many cases, the cases that end up being factually wrongful convictions are overdetermined. There's more than one cause, and if you took one of those causes away, they probably still would have been convicted. On the other hand, that is not the strongest argument for saying that the maybe secondary contribution of flawed forensic science gets entirely off the hook in that case. And there are many examples of that. But I would point out that there are examples in which virtually all the evidence was from flawed forensic science. Ray Crone, well, no, this is a Ray Crone case. It was a bite mark, which is just wrong.
There were two odontologists,
okay.
reputable odontologists, who said that that wasn't the guy. He was excluded, and then they went and they kept going down the line until they found somebody to say.
Well, I don't.
We don't.
I'd love to grab a beer with both of you after
my point, there's a second one. You say that people who, in visual hair comparison, never testify that way. I've got a tape recording, a videotape recording of the testimony of a visual hair comparison person who gave testimony in an exoneration case from, I think, West Virginia. I think it was from West Virginia.
I wonder who that was.
Michael,
the point is, she said it was almost certainly came from that particular person. She didn't testify in the terms you said.
Thank
So it
you.
was there.
People got beer tickets in their pockets. JoAnn wanted me to ask about error rates, but I'm not going to go there. So it's 5 o'clock. We appreciate your interest and attendance. Thank you very much for your questions. Thanks again the panel for